This is a protein. It is a transport. No, not the yellow submarine! Tissue repairment. Creation of hormone and enzymes. You heard it. That's right. They are very important biological molecules. You don't want to make Gollum feel depressed when my precious is gone forever. We need to make sure Gollum feels happy. Now the protein is made of 2 stages. Transcription from DNA to RNA and translation into a polypeptide chain. Alright, we don't have the budget to explain the next part, but we will leave you with something. This occurs in the cell's nucleus where the DNA is housed. Except, the DNA is not the golden idol. It's even more bloody precious. It is a child's dream to see this come true. It's Willy Wonka's recipe book. That's right, you heard it right. It's the treasure that Indiana Jones should be seeking. In that book, there are certain recipes that no one else could create, and those recipes are the genes. Those genes are going to help produce mRNA, which are the silver lightning chocolate. This mRNA is ready to travel to ribosomes to produce proteins. In a minute, you will know what those are. Of course, everything has a process, and for transcription, there's 3 stages: initiation, elongation, and termination. Just imagine Robin Williams as Willy Wonka for 1 second. Oh boy, wish that happened. Now, each one of his students is RNA polymerase. They will bind to a region of those recipe books called the promoter, and that is the start of RNA synthesis. There's a section that Wonka wants to keep for final publication. The other section is collecting dust in the bin. These 2 sections are the double helix strands. The one he uses is a template strand, and the one he separates is the non-template strand. From this point, RNA polymerase will start separating the double helix DNA strands and adding complementary nucleotides to the template strand, which is the DNA that is copied. He's composed of guanine, cytosine, adenine, and thymine. The guanine is stuck with cytosine and adenine with thymine. During elongation, the RNA polymerase moves along the DNA chain, joining more nucleotides to the mRNA strand. The complementary mRNA is produced from the base pairing rules, except that uracil replaces thymine and is the complementary base to adenine. 3 of these nucleotides make up a codon, which will later code for 1 amino acid. During termination, the RNA polymerase reaches that
"You are terminated,"
tells the polymerase to stop. The mRNA leaves the nucleus and enters into the cytoplasm. This factory is the ribosome, and that is made of rRNA. This serves the site where translation can occur and serves the chocolates. And you guessed it, the proteins are indeed the chocolates. Did you just hear that? Those are the Oompa Loompas, and they are certainly the creators of the chocolate. Those are the tRNAs. Every special recipe will involve secret ingredients. As you can see what the Oompa Loompas are doing, they're holding those ingredients, the amino acids. The Oompa Loompas don't have the brains to figure out what ingredients need to be added to create chocolate. So they eat the silver lightning chocolate and get inspiration. Each mRNA, that chocolate, has a codon, a 3-letter RNA nucleotide code, which corresponds directly to the anticodon on the tRNA. The anticodons are the cogs in the Oompa Loompa's brain that allow them to create the chocolate. The hands, which are amino acetyl tRNA synthetase, are the proteins responsible for attaching the amino acid to the tRNA. The starting codon is AUG, with the Oompa Loompa's brain cogs producing the message UAC with the corresponding amino acid, our ingredient meth. No, not that meth! Yes, methionine! Once the tRNA with an anticodon UAC binds, the first amino acid of our protein has joined and kick-started the whole process. This is called initiation. The nucleotide bases are added again and again and again. The Groundhog Day loop has begun. The amino acid the tRNA brings forms a peptide bond with methionine, extending the polypeptide chain. This is elongation. As the ribosome continues to move forward, the number of amino acids increase. But
Nobody's perfect.
as one could imagine, the Oompa Loompas are not perfect. Here we shall see them in action.
Oh no. You gave me the wrong nucleotide. We're getting a point mutation. Inspector, come here.
Uh-huh. This is silent. I don't see any changes.
Stop it! Inspector!
You're creating a missense mutation. You're changing an amino acid and creating a chocolate that Wonka will not be goddamn happy with.
Not again!
Since the mRNA structure, whose codons are used in translation, are taken directly from the original DNA molecule, DNA is the ultimate director for protein synthesis.
That's all, folks!